All right, so continuing on, we're just going to finish up our anatomy of the neurons because those are our most important cells, right? Certainly the neuroglia are going to be helping along the way, but the neurons themselves are doing the job of communicating and controlling, etc. So one of the things we were saying is we talked about the output part of the cell, the mouth part of the cell was the axon, and the axon can be myelinated or not. So remember that myelin is going to be basically like a lipid-based material that is an insulation, right? Just like maybe you have insulation along you, or wires have insulation, right? So that the signal doesn't go different places. It makes the signal, if you will, or the electrical impulse go faster. Same thing in terms of our axons. So it protects and electrically insulates our axons. We're not getting crossed wires and things like that, and it increases that speed. So what we find is that if we have neurons where the axons are myelinated, they're called myelinated, right? They have the myelinated sheath, but there's also non-myelinated ones as well, which are going to be slower. So it could be that the non-myelinated ones have less important information, or they're very short. So why do we need to go very fast? Because they're not going long distances, things like that. Now remember, we had two different ways of myelinating our cells depending on whether we're in the central or the peripheral nervous system, right? The central nervous system were insulated by oligodendrocytes. The peripheral nervous system, they were insulated by Schwann cells, and that's what we see here, right? So a Schwann cell grows, and we can see the cell nucleus there. The whole cell itself grows around the axon and insulates it. And so it's kind of like cinnamon roll around the axon, and it's also layering in that myelin as it's growing around it like that. But remember, it's only doing it in distinct portions. It's not continuous like a regular wire. So there are these areas where there are gaps. There's exposed wire. And in a regular wire, that might be a bad thing, but in a neuron, it's very important. So the myelin sheath gaps occur between the Schwann cells, and these are called axonal nodes or nodes of Ranvier. And so those are going to be very important in terms of conducting the impulse down those myelinated axons. So then the non-myelinated fibers, they're also surrounded by Schwann cells, but they don't have the coiling, and we don't have that whole myelin going around it. So the cell that we see here, our example cell that we saw before, does have myelination, but again, you can see those gaps, those nodes of Ranvier that exist between it as well. And so what we're going to find is the signal kind of hops along, if you will, those gaps in a general way. So in the central nervous system, remember, it's the oligodendrocytes that are producing that myelin sheath, and they are connecting a bunch of different cells at once, up to 60 different axons. Like we said, very complex. So when we look at that, it's still going to produce those axonal nodes, etc., but it's going to be this very, very complex network of oligodendrocytes with lots of different neurons. So in any case, that fat, if you will, that protein or lipid-based material that is wrapping the axon, if you think about fat, like if you went to the meat department and you looked at a steak or whatever, and you looked at where the fat is, what color does it appear? So the muscle, which is the meat, appears red, but the fat appears white, right? And so what we find is when we have a bunch of neurons that are insulated with that lipid-based material, those neurons, and then nerves, if you will, if we're bundling them all together, look white. So when we talk about gray matter and white matter, the white matter is where we have insulated axons that are all myelinated, and then gray matter is where we don't have them. Usually we're just going to have cell bodies and then non-myelinated fibers instead. So think about like the cerebrum. Most people know this is the cerebrum up here, right? Along the superficial surface of it, you have that gray matter, right? The cerebral cortex. That's where you're processing, just communicating between cells inside of the brain. So we're not going long distances, so we don't really need those to be myelinated. Meanwhile, if you're going down through the spinal cord or up from the spinal cord or up from the limb or something like that, those are long distances. We want to make sure those are myelinated. Now, distance isn't the only reason, but it is one of the important ones. So there's our oligodendrocytes again, insulating multiple different nerve fibers. Okay, like we said, in terms of the nerves, And the neurons making up the nerves, those neurons all have the same job to do, communicating signals, etc. But they're grouped in two different ways, structurally or functionally. You definitely need to know the three different structural groups and the three different functional groups. Important. Okay, so structural, based on their anatomy, right? So they're multipolar, they're bipolar, and they're unipolar. So multipolar are like the ones that we've been looking at already as our example one axon, and then many other dendrites. So this is the most common one. It's kind of like when we use like a femur or something like that for a generic bone, but there's lots of other bone shapes, etc. So multipolar is the most common one, and it's very common in the central nervous system where we have to make lots of connections, but there are also bipolar ones. So bipolar ones, like we'll see here, right? So here's our multipolar one that we saw before. Each one of these little extensions, dendrites or axons, counts as a pole. So when we look at a bipolar one, it doesn't mean it's like happy and then sad or anything like that. What it means is it's got two poles, one here and one here, right? So two different poles. We got one axon coming in, dendrite coming in, and one axon going out of that cell body. So we're just counting the number of poles off of the cell body. So that's bipolar. Unipolar, the cell body is off to the side. There's a little extension or neck to it like we see right there. And then we've got our communicating parts of the cell. But because there's only one pole coming off of it, we call that unipolar. So bipolar ones we find in unique places like in the eye and the olfactory. And then the unipolar ones, or even they call them sometimes pseudo-unipolar ones, these ones are usually associated with like sensory receptors, etc. But they're very specialized as well. Okay, so those are three structural types, if you will, of neurons. So multipolar, bipolar, unipolar. Now, functional classes of neurons means they're all sending signals, but where are those signals going? So what we find is that there are sensory neurons. So we said sensory is afferent, right? This is where the impulse is coming in. So these are neurons that are relaying the information like from the body towards the central nervous system, right? And most all of these are unipolar. Remember we saw unipolar already? So unipolar, those soma or cell bodies will be gathered together like in ganglia, and the signal is going to be going out right from your fingertip or whatever and routing up to the spinal cord or something like that. So usually, like we said, the cell bodies are located in ganglia in the peripheral nervous system, but they're routing that information to the central nervous system, bringing it in. Then there's motor, right? Motor is the opposite. Sensory afferent, motor efferent is going the opposite way. So from the central nervous system to the effectors. And these are usually multipolar, like we said, having to communicate with lots of different cells. And the cell bodies are usually in the central nervous system. And the last type are going to be what are called interneurons. Now, inter, remember, means between. Just like we talked about the intervertebral foramina, the holes in between the vertebrae, etc. Interneurons are also called association neurons. They are communicating between the sensory neurons and the motor neurons, right? These are the processing neurons, the ones that are thinking about it, deciding what's so important about the sensory information and how do we need to respond to it. And so those are the ones that are entirely within the central nervous system, the brain and spinal cord, and make up 99% of the body's neurons. That's the processing, that's the deciding, that's the thinking. Once you do the relay, in or out, those things are fairly simple. But all of that we're talking about up here, interneurons. Okay, so that finishes up our tour of the neurons themselves, right? Different parts of an individual neuron, and then different classes of neurons based on either their structure or their function. So we're going to leave that as a nice short little finishing touch to the anatomy of the neuron before we get into the physiology of the neuron, which has to do with, right, the molecular level, right, down at the chemical level instead. So that's always a tough one, so we're going to stop right here, we're going to pause right here, and we'll start with that physiology of the neuron coming up next.